work, and he just defended his PhD. We add strains. What we did was we tried to optimize strains that from the Asian lineage and the African lineages, and then design the primers to see if we can get new lineages within that population in Nigeria. And we got interesting ones. And we sent this, contributed it to the Genomic Bank, and has been deposited with Ascension number written. And it was tagged Ilorin Zika virus strains, which is a new strains already in circulation in Nigeria. Next slide. Microbiology, biotech, SDG 6 is also one area I promote a lot, which has to do with clean water and sanitation. I have done extensive work with my group, particularly using bioflocculants in wastewater treatment. We have reported extensive work on this, and I've reported 19 new strains to the NCBI and a session number given to us, which are completely new, not reported anywhere in the world before. We have characterized these strains. These strains of this have been characterized, and we are also harnessed the flocculant from these strains. One of the works is what I'm doing right now as a visiting professor at the University of Warwick in Coventry, United Kingdom. We have sent these strains now. We are about patenting 3 of them with the Warwick Ventures, and I've sent 4 of these strains to NOTAP in Abuja for patenting. We've discovered that these strains, particularly the products from these strains, now can act far better than alum and ferric chloride in terms of flocculation. In fact, initial study that we just finished, we saw that this extract actually has 97% what we call removal efficiency of biological oxygen demand from wastewater. Quite high, quite high. This is fundamental, and I must tell you, this is driving most of the researchers crazy over the world today, looking onto that, that product very soon that will showcase Nigeria in the global space. We have also worked with composite materials in the past. We've designed some composite materials. Let's move to the next slide quickly. Next slide. The composite material which we've designed has also helped us also to do some certain works which we tend to remove viruses and bacteria from water. We use nanoporous composites with some of my colleagues from Redeemers University in chemistry department, and we are able to establish that with those nanosite composites, we're able to actually remove bacteria from water which has been polluted with some infectious materials. So this is the bioflocculant. This was announced when we had this contribution, and this is the composite materials which has been, all these have been published actually in scientific papers. Now, next one, next slide. We got a grant from Third World Academy of Science. That's what we used for this study, for the nanocomposite. We developed a novel meta-doped pathrostatic hybrid clay composite for points of use of disinfection of water. These have been annexed. This is being used in some rural areas now, particularly in our local communities, for clean water supply for the populations that cannot have access to clean water supply. Next slide. The prospect and future outlook, as I'm rounding up, biotechnology is poised for an exciting future characterized by transformative innovations and groundbreaking advancements. The prospect and future outlook about it are promising. I must say they are very promising. I just shared some few things we have done, which is already pioneering a lot of things in the world now. As I said, 3 of those products are being patented now with United Kingdom, and 4 already sent to Abuja for patent, driven by various factors shaping the landscape. If only we can solve those things. What are they? Number 1, synthetic biology innovations. Let's move to the next slide quickly. These synthetic biological innovations, this involve engineering microorganisms with biofuels. Professor Charles Adetunji has done a lot of work on this, biofuels production, to environmental remediation. These are areas which people are really working on in terms of biotechnology now. Next slide. Biotechnology in healthcare advancement. Of course, most of our senior colleagues here, Professor Loke, has done a lot of work on these technologies like Cripsins gene editing, used purposely for some modifications in terms of DNAs so that we can advance our areas in vaccine development. These are critical areas which we have also used biotechnology. And then what about the global collaborations? The global collaborations are there. These are encouraged international collaborations. In the past, we have nothing to do with these guys out there, but now they have a lot to do with us. In fact, they cannot survive without Africans now. I think we need to clap for ourselves as Africans. They can't survive without us because the rules is clear. They can't come here. Even if they want to come here, Boko Haram will deal with them. So they will prefer to use those who know what Boko Haram names. So they can't come. So we have the space now. We are dominating the space. We're in control. So and that's the essence why I'm just telling you that the world is a better place for every one of us now as scientists now. Next slide. Artificial intelligence. Look at artificial intelligence. This is one area, integration. Currently now, we have a group now which we are trying to develop a proposal at United Kingdom, looking at AI in terms of wastewater surveillance and infectious disease control. We are trying to merge three aspects, how we could use AI for sample collection. AI is the in thing now, globally, and everybody is really interested in AI-driven analysis for identifying potential vaccine candidates in particular. Accelerated discovery, like the mRNA vaccines, which was produced not too long ago. Integrated global surveillance. These are pandemic preparedness, which I just shared some of the things. I've been able to share with you new genotypes, which biotechnology have been able to help us to discover, so we can prepare. So if I was talking last week now at the Abuja now, the minister was there to tell them, yes, good, we are celebrating, but we still have a lot to do. We are celebrating, we are rolling out HPV vaccine. But we need to know that there's a need for us to start vaccine production for Nigeria, because we have some new strains, which those who take those ones might not be able to cover for them. So these are areas also. Now, thermostable vaccines. Our condition in Nigeria, we cannot wait to continue to receive vaccines from international world, from Gavi. We need to produce ours with thermostable vaccines. Vaccines that will be overcoming storage challenges, where you can take them to rural environment where the roads are bad. I've traveled almost every coast of this nation as a national coordinator of poliovirus containment. And I've gone to all the zones. There's some places you have to be on bike for 8 hours. There's some places you have to be on boat. And these are places vaccines must get to. Can't we begin to talk about thermostable vaccines that does not need all these cold chains we are talking about? These are the areas biotechnology can also help us to enhance. Future trends in vaccine, R&D. Let's move on to the next slide. The future trends also. We can begin to look at advanced vaccine technologies. Look at what brought these two guys, these two Nobel laureates, Katali and Westman. They came together to collaborate. They are from different specialties. And they brought in biotechnological techniques. And today, they got the Nobel Prize for mRNA. And what mRNA has done for us is to let us know that we don't need to wait for 10 years before a vaccine can be developed. In other words, look at the time space that we got mRNA vaccine. Shorter period, because they bring in technological techniques that brought about prompt, speedy development of a vaccine, which was actually earned them a Nobel Prize. What about vectors-based vaccines are also there? We should begin to look at what is called personalized vaccine. These are areas we want to begin to think about using biotechnology. It means using genomic medicine, where you can actually tailor vaccine based on individual genetic profiles. You can profile individuals and then tailor vaccines to them. These are what we think we can do biotechnologically. So we have a lot to do to collaborate together. Chemists, microbiologists, biochemists, every one of us, we have a lot to do together. And then, as I close, what are the challenges and objectives? Problems, opportunities are there. The challenges are ethical concerns. We need to get to a point where we must agree that we should stop cutting corners as researchers. Ethical concerns. Why are people rejecting our vaccines? It's because many of us, they know how we behave. They know some people are not trustworthy. And then you that they know that you are not trustworthy, they say you develop a vaccine in your lab. They say, who? They now mention the name. Umang Parogo. When you hear Umang Parogo, only I can't take it too. I can't take his vaccine. Because already the ethical concerns are issues. Today in Nigeria, if you give me water now, I have to look at it twice before I drink it. So we must get to the point where we must talk about even regulatory hurdles. Today, some products are bad, and that's why whenever I talk, whoever is there, we just want to talk, agree to ourselves to go back and do the nitty-gritty. Let's go back to the drawing board. Because if something is not okay, why do we say it's okay? These are things. As long as you have dropped an envelope for the person, you say, oh, it's okay. Yet he has not tested it. You say it's okay. So regulatory hurdles are there. Potential misuse, these are all there. When we said there should not be COVID virus in the community in Nigeria, when we were carrying out this study, I was the national coordinator of polio containment. We were surprised to find vaccine virus. We saw it in cosmetic shops in Nigeria for the first time. It was shocking to the whole world. We saw vaccine virus in cattle, what do they call it now? Cattle markets. We asked those cattle markets people, how did you get this vaccine virus? Vaccines. This measles, all these things, polio. You know what they told us? It was surprising. The cattle man told us, he said, those vaccines, when they give it to their cattle, the cattle is bigger. In Nigeria, they use vaccine for cattle. So they will buy the bulk from people, give it to them in the hospitals. And people are looking for vaccines. And then they go and give it to cattle so that the cattle will be big and bigger, and then they tell you to come and buy it for 500,000. So this is the kind of things. We saw it, and we reported it. And you know, we reported it as national coordinator. I did. And that was one of the things that Nigeria was actually commended for, because we went drastically to fight those people who are using it for capsule vaccines. Because we know what the implication is. They are using it for their capsules because of their own financial gains. People who are buying the capsules, what happened to them? We don't know the end-time implication of people, the health implications that go to those people. Now, solving complex problems, that's the opportunity of biotechnology. Advancing research is also there. We've advanced a lot of research with biotechnology in terms of, and then addressing global issues. And then in conclusion, next slide, I like to say, distinguished ladies and gentlemen, that bridging the gap in biotechnology research applications is a critical endeavor with far-reaching implications for both scientific progress and societal well-being. If for that alone, I think I'm comfortable to be a teacher and a researcher in Nigeria, even though the pay might not be good. But the truth is, for the society well-being, gives me joy to keep on going. And how do we do this? We are all in the same situation. Look at that man. He's trying to climb to see outside there. And but do you see what he's climbing on? These are ladders. Ladders packed together. He did not know how to do what? To use it. He just put the ladders and he was trying. Only 2 of those ladders will make him to do what? To see. And we have packed of resource people in Nigeria, intellectuals, people who have all it takes to revolutionize this country. Just earlier this year, I was endorsed by the Royal Society of the United Kingdom as a global exceptional talent. And what it entails is that I have access to everywhere in the United Kingdom. And I asked myself a question. Royal Society of the United Kingdom endorsed me as a global exceptional talent. Nigeria Kingdom has not endorsed me. It's interesting. It's so interesting sometimes. We have all it takes here, but we don't respect what we have. So all the ladders are there, but we are still struggling with infectious disease and still thinking about what to do. And then finally, look at that man seated. I don't know who the man is. I decided not to use a black man. It must be a white man now. Because a black man, we are finding our way now. We are getting out there. The man is seated confused. He has all the tires, better tires on top. But look at the only one tire. And then he could not even use the one tire he has there. Ladies and gentlemen, on this note, this is me. Next slide. This is all I do. Can we go to the next slide? It's me. It's me there collaborating with my colleagues in the UK. Me on a national assignment. I go to rural areas to do some rural assignments. And then me with the Minister of Science and Technology when I was actually patenting one of the products. And then myself also with some national assignment. And then also receiving a plaque. And then, of course, my collaborators in Warwick. And then, of course, some of my PhD students. I'm happy to inform you that 3 years ago, as you read from my citation, was 3 PhD graduated, but today I've graduated 10 PhDs. Thank you very much. And these are some of the PhD students, and these are myself, enjoying myself in my house. I also relax sometimes, even though I work with viruses. And then I play football too. Can we, and so that we cannot run to. And then thank you. I want to say thank you again. Next slide. Keep in touch. These are my emails, address, and my phone numbers. And then, of course, acknowledgement. Next slide. I'd like to acknowledge all partners, particularly University of Lawrence, TUAS, Imperial College, Wellcome Trust, TEDFund, World Organization for Ministry of Health, CETA, Warwick, and then NYU, New York Universities, and of course, thank Precious Cornerstone University for hosting me again this year. On this note, I say thank you very much. God bless you. Thank you for listening.
Thank you very much. Yes. Thank you very much, Professor Kolawole. Can we appreciate Prof again? So it's time for questions, comments from our participants. All right.
Thank you very much for your presentation, sir. I have 2 questions. The first one has to do with using tools, GIS and remote sensing tools for surveillance. So I want to ask, how can we use these tools to achieve One Health? Because I know that's where the world is going. especially as you talked about SDGs 2, 3, 14, and the likes. Then secondly, you mentioned nanocomposites, that is for their use in biotechnology. So I want to ask, and there's been reports about their health implications, especially as they are reported to be cytotoxic. So how do we use such material, since we want to manage the environment so that we are not introducing something that is also toxic to the environment? Thank you so much.
Thank you very much. Can I take it? Oh, thank you very much. That's a beautiful question. Please, can we just clap for her too? Thank you very much. I start from the last one. The last one has to do with, that's the interesting thing. The research is continuum. Somebody did a research and said it's toxic. That means that I need to go and also do mine also to check that there's a way I can improve on it. So I want to say that, yes, there are limits to absolutely nothing you can say that is not toxic in life. Even water could be toxic. Vaccines, some have said, oh, it's toxic and all are like that. But the truth is that we are talking about if you can do what we call a kind of review on some of the components that we use. I'll give you an example. The bioflocculant we are talking about now, the advantage of the bioflocculant over the composites, as I talked about, we have worked on composites. Yes, it was active, good, but it has its own limitations of toxicity. That led us to think about microbial agents, and we're able to extract bioflocculants. The composites, the chemical synthetics, chemical flocculants, they are actually not eco-friendly. But the interesting thing that is making this bioflocculant to sell now, which people are running after us now, is the fact that it is biodegradable. It was because of the challenge of the fact we got with the nanocomposites that made us to start thinking out of the box. To see, yes, nanocomposite is good, but what about the environmental challenge? So we now went further to begin to look at other things that could be environmentally friendly. As I speak with you right now, the advantage of bioflocculant over the nanocomposite is the fact that it is biodegradable and eco-friendly. It doesn't behave like that of the nanocomposites, like the chemical flocculant, like alum and ferric chloride. If you use it for wastewater treatment, for alum and ferric chloride, it becomes a nuisance within the environment. But the one we are talking about in the bioflocculant, when you use it, because they are products from microorganisms, the moment they have treated it, the flux just degrade. They are biodegradable, and then they just move out of the environment like that. So I will actually say, to answer your question, that you even when you have done something and they say, or you did it, it has toxicity, don't stop there. That's what they call research. Don't stop there. Keep moving. And then, of course, for everything in life, there's always the limitations. But when you have more advantage over your limitations, it shows you that you are making progress with this. Okay? I don't know if it answers that one very well. Second has to do with the GIS. Thank you very much. That's a very good one. I love this so much. The SOMAS, have you heard about SOMAS? SOMAS is what NCDC is using. It was SOMAS that was developed when COVID came. SOMAS means that you can report our tested samples anywhere they are in the labs and submit it to the system directly, while the NCDC will just get it and then analyze and know what's the trend, what's happening in Nigeria. Success of SOMAS or GIS is now what we are leveraging on the new grants now, which Nigeria again has just gotten in terms of environmental surveillance by the NCDC. I'm happy to inform you that just last month, November, Nigeria has just won a new grant, which by the grace of God, I'm also one of those who is also in that team with the NCDC. We got a new grant from Bill and Melinda Gates that will be pioneering environmental surveillance in Nigeria. You want to clap, you can clap for Nigeria. Nigeria is doing well. Nigeria is doing well. And I think we can clap for the DG of NCDC also, Dr. Adetifa. He brought myself, he dragged me and Professor Iruka Okeke in UI. Both of us were the consultants who worked on that team with the AFENET and NCDC. And by the grace of God, last month it has been announced, Nigeria just got the grant to pilot environmental surveillance in terms of clinical diagnosis in the country for all infectious disease, so that we can now begin to look at other approaches to be able to know what is happening in our communities. So SOMAS is what we deployed. In fact, when we were reviewing final stage, we were asked which tool are we going to use. We looked at it as SOMAS has been used successfully, so we are bringing SOMAS again for this one now, so that what we'll be having in the whole nation now, so much will be used for that approach. So the GIS has been a good thing. So that's why I said we should also begin to think about GIS, AI, so that we limit our stress, and then we can leverage on our existing success and move forward to predicting what we want to do in the future. Thank you very much for the question. I appreciate that. That's very good. Thank you very much.
Please appreciate Prof for the response, powerful response and presentation. Any other question? Okay, thank you. So next on our agenda is to take the debate for our schools. Hello, students. Hello, students. Please, can I have 2 students from each school? Please. Hello, it's time for your debate. Please, can you come forward? Please. Okay. Just one school. Okay. So your name, you and then 1 minute question. Two of you answer.
My name is Afolabi David. I'm in SS2 from Christ Life College.
My name is Ogini Bukunlua. I'm in SS2 class, Christ Life College.
Thank you very much. I have just 1 minute now. You answer this. What is biotechnology? Okay, the next question is, what subject in your school is related to biotechnology?
Biology.
Biology and chemistry.
All right, a round of applause for them. You can go. So we'll be moving straight to our keynote speaker, Professor Thomas Webster. He'll be presenting online, but before he will make his presentation, I will read his citation. Professor Thomas J. Webster, degrees are in chemistry, chemical engineering from the University of Pittsburgh, and in biomedical engineering from RPI, USA. He served as a professor at Purdue, Brown, and Northeastern University from 2012 to 2021, serving as Chemical Engineering Department Chair from 2012 to 2019. He formed numerous FDA-approved medical products, currently improving human health. He is currently helping those companies and serve as a professor at Hebrew University of Technology, Savita University, Valori Institute of Technology, and others. Professor Webster has numerous awards, including 2020 World Top 2% Scientist by Citation, 0.1% most special researcher in pharmacology and toxicology, 2022 best material science scientist by citation, and is a fellow of over 8 societies. Professor Webster is a former president of USA Society for Biomaterial and has over 1,350 publications to his credit, with over 53,000 citations. He was recently nominated for Nobel Prize in Chemistry 2023. Please permit me to welcome Professor Thomas Webster as he makes his keynote address. Thank you.
Hi, can you guys hear me? Hi, if you guys can hear me, just send me a quick chat note to say yes. Oh, excellent. So you can hear me. That sounds great. So also just let me know on the chat function, how long do you want me to talk? Because as you know, I could talk forever, but obviously you have an agenda and a schedule. So let me know how long you want me to present. Great. So about a half an hour, maybe some minutes there for questions. That sounds great. So obviously, I wanted to be there in person. I think, as you all know, I don't know what is happening with the visa process. I guess it's been maybe close to a week and a half or 2 weeks that I'm still waiting for my visa to be processed. So it will be processed and awarded at some point, and I do hope to visit everybody in person at that time, of course. So I wanted to give a presentation, something a little different for about, but I'm also very interested these days in artificial intelligence. In fact, as you know, probably as was stated during my introduction, I have been very entrepreneurial. Here's a lot of the companies that I have spun out throughout my career. These days, at least in the United States, there's a lot of investment in artificial intelligence. So I wanted to describe in this brief talk how I'm thinking about using artificial intelligence in biomaterials. But before getting too far along this road, and I call this the Band-Aid. So here's your Band-Aid for healthcare, where the AI stands for artificial intelligence. Before talking too much about this concept of a Band-Aid using artificial intelligence to improve medicine, I wanted to announce something pretty important to me, and I hope pretty important to you. And obviously, I wanted to be there in person to talk more about this, but I'm super excited to announce this new fund. And really, the idea behind this fund, which is primarily being supported by investors, by my startup companies, by people I have known for generations, is to support people from Nigeria, students, whether you're a high school student, we have programs that can help high school students, all the way up to graduate students. We haven't thought too much about faculty, but maybe that's something we can consider. But supporting students who are interested in biomaterials from Nigeria to come to the United States, to universities, to conduct world-class biomaterials research. So this is not supporting students to come to labs to repeat experiments that somebody might have done, or to just get a sampling of research. This is real research in biomaterials. So be announcing more details as we get further along, details, logistics with you guys. But really the idea is that we fund that is going to support students to come to universities in the US to do research with me or other people, to learn about really world-class biomaterials research and have you contribute to the future of this field. So really, here's some of the details, but again, I really want to come there in person and discuss these details more thoroughly before we set up a website, before we set up an application process. But rest assured, this will happen. Of happening. So something that would really help me at some point during your conference, take a photo of everybody at this conference and send it to me, and then I can send it to some investors to contribute more money to really enable this program to occur. So I'm super excited to announce this. It is going to happen, and we just need to figure out some logistics. Hopefully, when my visa is issued, I will come in person and talk about this with some students, some of your faculty there, to really come up with the best program possible. But I've done this in other places where I have used funding from investors that I know, from companies that I know, to really enhance a learning experience for students in this biomaterials area. Okay, so that's the big announcement. Super excited about this, but let me now talk about the research, some opportunities that you could have if you participated in this program. And I need to give disclosures, of course, some disclosures in this area that have to deal with companies that I've started, that I have a financial interest in, this area of AI intersection with biomaterials. So, as you know, my last name is Webster. So there's a very famous English, US English, American English dictionary that's called the Webster Dictionary. So I think what that means is built into my genes, into my DNA, are definitions of new fields. So here's the definition, the Webster definition of artificial intelligence. So I think, as we all know, artificial intelligence is really the perceiving, synthesizing, and inferring information through a computer, not through our brains, a human brain, but really demonstrated intelligence through computers, algorithms, software, really artificial means to interpret health data if you're using AI for the healthcare system, or if you're using AI to drive your car, then it's using artificial intelligence for automobiles. If you're using AI to write a term paper, which I hope you don't, but there are programs called things like Chat GPT, where you can use artificial intelligence to write essays for you. So really, it's this whole idea of using computers to perceive information, to synthesize information, and infer or predict information, as opposed to using humans or other animals. So why do I want to consider using AI for healthcare? And there are really many reasons. There are many things wrong with our healthcare system that could be helped through the use of computers, software, algorithms to understand your healthcare system, your healthcare data, and predict how you might respond to a particular treatment in disease treatment or healthcare treatment. So here's a lot of things that I think are wrong with our healthcare system that could be helped through the use of artificial intelligence. We are all well too aware of the failures of our healthcare system in COVID. And I think what COVID did was not only highlight problems in our healthcare systems, but it also highlighted our failures to predict how you might respond to a virus. And that's where I see AI really fitting into our healthcare system. AI helping us get away from overcrowded hospitals, which did not serve us well during COVID. AI helping us reduce the barriers to get proper healthcare and predict, again, how you would respond to a vaccine or how you might respond to a medical device or an implant. Really helping us understand how you personally might respond to a particular medical treatment. AI, I think, can help us avoid medical device failure. We have medical devices that fail all over our body, and it's very specific to the patient. You might have a medical device, and your body might accept that medical device. It wouldn't wall it off with chronic inflammation. You might not have infection, but I have a different immune system, and maybe that same medical device does not perform as well. in my body as yours. Wouldn't it be great to have AI, artificial intelligence, predict that for me, that medical device is going to fail. For you, it's going to be successful. We also tend to treat every patient the same. We have a reactionary healthcare system rather than a predictive healthcare system. All of these things, all of these problems in our healthcare system, I think can be solved by artificial intelligence. So let me walk you through a couple of quick examples of how I think artificial intelligence will revolutionize healthcare. And let me start with examples from my own research group, examples from my own companies where we have developed static implants. And I think clearly you'll see that what I have done in my career so far does not embrace artificial intelligence. It does not use computer algorithms to predict the future. And I'll end the presentation with some examples of how we can use AI to really improve medicine. But traditionally, as you might know, I have a history of my lab group, my companies have a history of taking medical devices off the shelf, whether it's a spinal implant, pictured here as a 3D printed spinal implant, or maybe it's an amputee device. Here's a screw that is used to go into bone, and then an artificial implant is placed around that material. Or maybe it's taking a vascular graft, a stent, a material that regenerates the artery, or maybe it's a material that's used to attach soft tissue, orthopedic soft tissue, like ligaments, tendons to bone. Or maybe it's a catheter. Here's a picture of an inside of a catheter that we've modified to have nano textures. Whether you talk about any of these implants off the shelf, hip implants, spinal grafts, amputee devices, we have a history of nano texturing those devices to increase tissue growth, to reduce infection, inflammatory drugs, and we have seen this success. In fact, one of the things that I'm very proud of is we have 3 cases in particular of where we've taken medical devices off the shelf, spinal screws. These are called pedicle screws that if you have spinal fractures or a collapsed disc in your spine, we have taken medical pedicle screws and created nanotextures on those materials. They are currently in humans. In fact, about 14,000 of these screws have been implanted in humans today, and we don't see any cases of failure. No cases of infection when nanotexturing these screws in humans, no cases of extensive yes. So no cases of failure whatsoever when you these nanotextures. Can you guys hear me okay? Great. So yeah, let me know by the chat function if you cannot hear me okay, or obviously have questions as I go through this. We've also done the same thing for other kinds of chemistry. So in this case, we've taken titanium screws. We've nanotextured them to, again, have 0 cases of implant failure. And I should mention the industry standard for non-nanotextured screws, failure is about 5 to 10%. So the case that we're getting 0% failures is something we're quite proud of. We've also done the same thing, not just for titanium, but we've nanotextured other chemistries like silicon nitride, where we also see 0 cases of implant failure. And here's an X-ray showing some of those silicon nitride implants with nanotextures and have 0 cases of implant failure. We've also worked with another company called Tiber, where we've nanotextured foot screws that are used for diabetic. You might know if you have diabetes, you have an increased chance of infection because of all the sugar that is not metabolized in your body. That Those implants, again, are made of ceramics. We've nanotextured those. And again, we're seeing 0 cases of failure. So we have a history of really nanotexturing medical devices and improving their function in the body. The problem is, in all of these cases, this does not involve artificial intelligence. This is traditional implants where we've created nanotextures without using artificial intelligence, but we do see success. So we're very happy with this. But let me jump ahead to the presentation where really this nano-optimized approach is wonderful, but it does not employ artificial intelligence. In fact, here on the slide, we've trademarked this term nano-optimized, where we work with medical device companies. They give us their implant. We create a nanotexture. We give you a stamp that's called nano-optimized that can reduce or eliminate infection of your implant, eliminate chronic inflammation for your implant, and improve bone growth. Let me skip through these in the interest of time and really transition to artificial intelligence. How can we get from this moment where we've nanotextured implants without using artificial intelligence to now using artificial intelligence that can even improve these medical devices more? And this has to do with being more proactive, not just putting a screw in your spine, not just putting a screw in your foot and improving implant performance, but how can we use your health history to understand how you are going to react to an implant that's inserted into your body? And this has to do with sensors. I'm convinced that implantable sensors are the future, and implantable sensors will incorporate artificial intelligence to predict how well a medical treatment will give you to improve healthcare rather than this trial and error approach we have in healthcare today. So let me describe 2 approaches we're using to transform today's implants into artificial intelligence implants that can improve success for your materials. Now, we do have sensors in medicine. This is a pacemaker. A pacemaker obviously is put into your heart. It stimulates your heart to beat whenever it's not beating enough. That's obviously a sensor. It senses an event, responds to that event, and improves your health. We also have sensors for implant placement. So this green implant right here is used during knee implant surgeries. You can see it's put right here. It measures pressure and tells the orthopedic surgeon, has that knee implant been inserted surgically correctly or not? Of course, this sensor is removed later, but it uses pressure to determine if that implant is placed correctly for that patient. And of course, we have tons of sensors that we wear and can tell you your heart rate, can tell you your EKG, can give you your oxygen content in your blood, can do a lot of things to help your health. But this is not far enough, and it does not incorporate artificial intelligence. Here's the 3 circles that I think of for the future of implantable sensors. Obviously, you need something in your material to sense a biological event. We sense cell presence. Do you have bacteria present on your implant? Do you have inflammatory cells on your implant? Or if it's an orthopedic implant, do you have bone cells that are making bone? We develop materials that can sense that event. Then we send that information to a processor. And here's my iPhone. This is where we send information to an external device. And this is where artificial intelligence is incorporated. We have programs, we have software that interprets this information, whether you have bacteria, whether you have inflammatory cells, or do you have tissue-forming cells. It interprets that information with your healthcare history to predict if that implant is headed for success or is that implant headed for failure? So that processing part is where you artificial intelligence to predict how well that implant is doing. If you have a history of infection, if you have a poor immune system, you have an immune disorder, then that artificial intelligence knows your history and will be able to respond, cause that implant to respond to fight off that bacteria. In contrast, if you're healthy, and let's say your body can fight off that bacteria, it does not need any intervention, then that process knows that and it responds by not responding. You don't need any response for that implant. That body, your body will fight that bacteria and you have successful tissue growth. So let me give you two practical examples of how we've been creating these sensors, incorporating artificial intelligence to predict implant success or failure. The first we call the smart hip. And what we're doing is taking a hip implant, we are growing carbon nanotubes. These are electrically active materials. They can electrically sense what cells attach, how many of those cells you have, and where they are on the implant. And again, this could be a bacteria. So maybe this smart hip senses you have 10 MRSA, which are very dangerous bacteria to have in your body. You have that here on the bottom part of your implant. That information is sent to a computer algorithm where artificial intelligence will predict for you and your history, considering your immune system, and it will respond to fight that bacteria if necessary. In contrast, if you have a healthy immune system, that implant doesn't have to do anything, and it will just monitor those events to make sure that your immune system kills those bacteria and you do not get an infected implant. Here's some real data from an animal experiment where we implanted our sensor here on the right into the calvaria or the skull of rats. We were able to monitor bacteria presence, and we were able to on-demand deliver, use our cell phones to deliver an antibiotic to kill that bacteria. So in this case, we used artificial intelligence to indicate that bacteria are present and they're present at too high of a level to cause an infection and that your body would not be able to handle that bacteria. So we have the implant release gentamicin. It also released a bone growth factor to cause those cells through electrical signal. We grew using atomic layer deposition sensors off of implants. In fact, our animal data also is very promising for this approach. We had a sheep study where we inserted our sensor into the spine. Here I'm pointing on the slide to where these sensors were inserted. These sensors measured if infection was present, enabled the implant to release locally an antibiotic if that was the case, or no infection was present, allowed that implant to perform normally and in a healthy way. I don't know if it's possible for people to mute their speakers so that everybody can hear. That would be great. But here's digital health. So one thing that you might be aware of is on your cell phone, you can connect on your cell phone. And again, if everybody could mute, their speakers, that would be helpful. I'm not sure if I can do it from my computer panel, but here is an example where we are taking cell phones. Like this, thermal detector is called a fluor, can actually measure thermal profiles of your skin, of a wound. So here's an example of a patient who received that knee implant. Obviously, you can see the scar or the surgical incision. We can generate a temperature profile from that incision to tell us if infection is setting in or inflammation is setting in, or is that wound. So again, if people could mute. A lot of noise.
More baggage. Not plenty now.
That would be good. But let me continue. So as you can see from these images, we were able to get temperature profiles, and an increase in temperature can indicate if you have infection. An increase in temperature can also indicate if you have inflammation. So we are gathering this information and then sending it again to your iPhone to interpret that information using artificial signs of an infection, like we did here because of these temperature changes. We're able to interpret if that patient, considering their history of infection or not, can that patient deal with that infection on their own? Do you need to use antibiotics or et cetera, anti-inflammatory agents to ensure that that wound heals successfully? And we're using digital health, measuring temperature profiles of wounds. We're able to also do this internally. These are AI-generated images where we can look inside the tissue, measure temperature profiles to see if infection is occurring through those increases in temperature or inflammation.
Charlie, come Charlie, conseil distribution. Conseil distribution.
Combien là? This is an example of to internal problems in temperature. So let me give you the last example of where we're incorporating artificial intelligence. And very new information using 4D printing. I think as we all know, 4D printing is where you take a 3D material, you can incorporate a nanomaterial response to your and I happen to have subject the material to near infrared. You can see that red light coming off of the near infrared device. That near infrared will nanomaterial to change shape. So here's an example of where we 3D printed a flower, a hand, the brain, and again, using the near infrared system, this commercially available near infrared system, we were able to excite those nanoparticles and cause that material to change shape. So in the case of a flower, it can open up. We can control one petal at a time. The hand, we can control fingers to go up one at a time through remote, on-demand, near-infrared excitation. So let me show you how we're using this in medicine. Here's a video where we 3D printed a material. This has graphene oxide nanoparticles. You'll see a pink light that is shining on this material. You can see it reflected off of the surface. That pink light is going to heat up these nanoparticles and make it extend. So you see the material slowly extend because we caused those nanoparticles to heat up using near-infrared excitation. So we're interested in this approach because you can implant these materials into the body and cause them to change shape on demand and remotely. So things like scoliosis, curvature of the spine, we are inserting these materials, attaching them to the spine, and causing
Recording in progress.
Causing that material to straighten. So in turn, it'll cause the spine to straighten. We're also doing this, and I'll show you some results for healing intervertebral disc damage. So if you have a collapsed disc, you could insert this into the disc space, activate it with your near-infrared light source, again, like I have here. That can cause the material to swell and improve spacing between the discs so that patient doesn't have pain and is now able to return to an active lifestyle. We're also using this for sphincters, where sphincters get weak as we age, so you can use this material to open and close sphincters in the body to reduce acid reflux or reduce other complications. So let me show you some first-time data that I'm presenting here, animal experiments using these materials, where we first, I have two sets of animal data to share with you. We first inserted these under the skin of mice. Here's the near-infrared system. You can see the original shape of this material was curved. So that is what was originally inserted under the skin. Then we use near-infrared light to make it flat. So we know that this approach can remotely activate a material in your body to change shape. So here's where we're really getting to a real application. Here's two discs in your spine where we created a material that had a certain small shape. So that means we need a small incision into the spine to insert this material. Down here, I'm circling what it originally looked like. Then we use near-infrared to cause it to expand. That pushes collapsed discs away from each other. Here you can see how the shape can be expanded through near-infrared excitation. We also add a certain ceramic to strengthen this material so that it stays separated. And we have animal data in these rats showing that it does in fact bond to bone, and it creates a healthy way, a minimally invasive way to separate collapsed discs so that patient can return to an active, healthy lifestyle. So many applications of 4D printed materials. We are incorporating artificial intelligence into this approach because we're putting sensors into these 4D printed materials. And again, those sensors can communicate to a handheld device and with patient history, know if that patient has a history of collapsed discs. Do we need this to strengthen every couple of weeks, expand every couple of weeks to keep those discs from compressing, or is everything working fine? So we're incorporating computer algorithms of patient data, patient history, to know how well this material should work. So I hope in this very brief presentation, I was able to show you a couple of things. One, nanotextures work. And nanotextures can reduce infection without using antibiotics. They can reduce inflammation without using anti-inflammatory drugs. And they especially can improve bone growth. We now have human data supporting this. So we know over the past 5 years that we've been implanting these into the spine as well as into the feet, that incredibly, knock on wood, we have seen 0 cases of implant failures. This is in contrast, again, to industry standards in the spine, which have about 5 to 10% implant failures. So simply using nanotextures on current medical devices can improve and completely eliminate, we think, implant failures. But as I mentioned, traditional medicine does not use artificial intelligence. So I gave you 2 examples of sensors that we are developing that use artificial intelligence to predict if you're going to have a problem with that implant. And if so, we have created implants that, while they're in the body, can release an antibiotic, can release a pro-bone growth agent to ensure success. I also gave you a quick example of digital health, where you can use temperature profiles, these FLIR systems, you connect it to your cell phone, it can measure temperature profiles after you receive an implant. Those temperature profiles can tell you, do you have infection? Do you have inflammation? And then using artificial intelligence, again, knowing your history, your healthcare history, if you have a problem with infection, then we need to do something about it right now and keep that infection from occurring. If you don't have problems with infection, then let's just monitor that implant through that temperature profile to ensure that your implant is going to be successful. And then I ended with this idea of 4D printing, where we're creating materials that can respond to external near infrared. So if I had an implant in my hand, I could activate that implant using near infrared to change shape to improve device performance. Maybe it's treating arthritis in your knuckles and increasing or decreasing spacing between joints. Maybe it's straightening a spine on demand in a controlled way rather than receiving spinal rods repeatedly. Have one material that can straighten the spine, one surgery on demand. Or maybe it's treating that intervertebral disc collapse that can occur in your spine, cause that shape change to lengthen, and it can do it in 5 years, 10 years, 15 years without a new implant. So a lot of excitement, I think, using AI, using these brand new approaches to improve our healthcare system that, again, as I started the presentation, if you participate in this Nigerian-US program for research, you can be part of this research as well. So, of course, I want to thank the funding acknowledgements. I want to thank my lab group. Here's a picture of us eating at a restaurant. I have many books constantly coming out with new books if you're interested in reading more about this. And I'm super excited to announce this journal that I'm editor of. It's actually based in Kenya in Royal Publishing Company, where we're publishing open access for anything related to medical and health sciences. And here's my email if you have questions about the journal or questions about my presentation. But stay tuned, because hopefully when my visa is issued, I will be able to come to Nigeria and we can this hosting of students, paying for students to come to the United States to be exposed to really these future directions in biomaterials, sensors, 4D printing, artificial intelligence, nanotechnology, etc. So thank you very much for your attention. Again, I wish I could be there in person, but I know that's going to happen soon enough. And please email questions by the chat box too if you're interested. And I thank you for your attention. Thank you very much.
Thank you very much, Thomas West, for your presentation and numerous research about biomaterials. So at this point, we want to also Oloke to interact with the keynote presenter.
Yeah, thank you very much, Professor Webb Thompson. By the grace of God, next year, you will be coming physically to launch the foundation. And I'm happy to introduce to you, which will present you your gifts and everything, but because of you not present, when you come next year, that means you are going to take double award and gifts. But in order for me not to use stretch more effort and intensify more on the foundation that we'll be having in incorporating many distinguished professors, postdoc, scientists, at least minimum of 10 to 15 every year to United States of America. I will be in charge of the foundation by the grace of God, God helping us. And if you know that you need many of these network in US, you see reason why you have to join RAVE now. He would have been here today. He had support of many billionaires and many other things that have supported this foundation. I would have launched it here today, but unfortunately, so many of our guests have a visa issue, but it's not a problem. We will collate and start from next year by the grace of God. And for me not to talk too much, I will call our father, distinguished professor of eminence.
Thank you so much, Professor Adetuji. At this juncture, I want to appreciate our keynote speaker for a well to assist with students. Get in touch with you. God bless.
Any question? Anything on collaboration on the biomaterial science?
The presentation is very interesting and exciting, but my major question is about the collaboration. The question is that since the resource person will be coming next year, we wouldn't know if the conference is happening. I want to know how we can get involved and be informed when the collaboration will start. Thanks.
Thank you very much for your presentation, Professor Webster. My name is Bolani Akinsonla from Landmark University, and I'd like to ask as regards AI in healthcare. Can we begin to, is there a bother, or should we be confident as humans? Will AI at any point replace human when it comes to healthcare? Thank you.
Please go ahead, Prof, for your response. Professor Webster?
So, I think the conference is annually. We do it annually. So there is a platform we have, RAID platform on WhatsApp. You can drop your number with the registration desk. We had all of you on that platform. For you to be getting, we send updates every day about this, whatever we are doing, and it has been for the past 3 years. So we can add you more on that platform. So, and so many other questions can be sent, and so the network, by the grace of God, let's keep in touch so that we can move ahead. So let's run.
So we are closing now. Then our participants can go for their lunch, collect your ticket at the desk, and file to the lunch center for your meal. Thank you. Okay, there will be cocktail party after the lunch. Tomorrow program will start by 9 a.m. Professor Adetuji, 9 a.m. Technical section will start by 9 a.m. and poster. Thank you.